Hello, I'm Nishat Tasnim. Today I'll share my project with you titled Synthetic DNA Sequence Generation and Classification for Species Discrimination. This is our agenda. This project aims to generate synthetic DNA sequences through a generative adversarial network and classify DNA sequences using a random forest classifier, support vector machine classifier, and logistic regression classifier with KMR encoding. This project is important as it can generate new, high quality synthetic genomes with minimal privacy loss to solve the data availability problem. These models are also cost effective. Although next generation sequencing technologies have made it possible to generate a large collection of sequences quickly, current genomic data still suffer from small data sizes, imbalances, and biases due to various factors, including disease rareness, test affordability, and concerns about privacy and security. One of the machine learning problems that is difficult but important to address is imbalanced data, where data is recessive while the others are dominant. Most of the classifier's performance is significantly degraded when dealing with imbalanced data. To address these limitations of genomic data, we developed a conditional generative adversarial network to enhance the amount and diversity of genomic data by transforming samples already in the data rather than collecting new samples. The number of raw DNA data for rats was 2173 and we generated 827 synthetic data. The actual data was 705 for human and the generated data was 2295. In the case of rat dataset, the GA training exhibited a relatively lower discriminator loss of 0.81 and a generator loss of 0.91 after 200 epochs, indicating a good convergence. The average test loss of 0.75 for rat sequences suggests that the generated synthetic sequences align well with the actual rat DNA data. Conversely, for the human dataset, the GAN training showed a higher discriminator loss of 1.21 but a lower generator loss of 0.56 after 300 epochs, indicating some challenges in convergence. However, the average test loss of 0.75 for human sequences is comparable to the rat dataset. These results illustrate that the synthetic data generated by the GAN model can be effectively classified by these machine learning algorithms with SVM achieving the highest accuracy. In contrast, when the models were tested on actual data without synthetic data, their performance decreased, as evidenced by lower accuracies, imbalanced confusion matrices, and lower F1 scores. This underscores the importance of using synthetic data for training and testing when actual data may need to be more balanced. In future, we will explore fine-tuning of hyperparameters and architecture for the GN to enhance sequence diversity and quality, and we will investigate additional evaluation matrices and domain-specific measures to assess the biological relevance of generated sequences.